So today's lesson, we're going to focus on looking at the second line of defense. Although you guys have already covered it, make sure you're listening up as to make sure that you understand the mechanisms involved and also the processes that fall under the second line of defense. Okay, so just as a quick recap, the first line of defense was all about defense, right? Making sure either one, they don't get in at all, right? There's a physical barrier that's stopping it from getting in. Or second thing, there's a chemical barrier that's also preventing any microorganisms from getting a foothold in any places that they can potentially get in. And also third is a microbial barrier as well, which is basically talking about uh, microorganisms acting as a barrier because they are the native species on you, and they basically have set up shop everywhere they want, and they don't want any new competitors coming along. So that is also a barrier that acts on. Okay, so in this example here, right, when a bacteria comes in contact with your body, they must gain access to living tissues to become pathogens. List two possible routes the bacteria could use to access the living tissues of the body. Essentially, what the question is asking, when microorganisms are trying to infect us, they have to face the first barrier, our first physical barrier, which is all the places where our skin is preventing the microorganism from getting to our living tissue. But what are the possible points of entry that these microorganisms could get in? Okay, what's the answer? Abi, what do you think? Technically, you're the only one who hasn't answered this question. Everyone else has. So what do you think? What are the two possible routes that bacteria could use to access the living tissue of your body? Through eyes, possibly. What's the other one? What's another one? So rather than saying the organs, you should say the system they're part of. Okay, for eyes, obviously you just say eyes, but for the nose, it's more towards the respiratory system. That's how you would describe it, right? In fact, that is what the answers are stating, right? Eyes or the respiratory surfaces, or the most straightforward one that you guys should always link in your head is a cut in the skin. Okay, anytime there's been a breach of the first line of defense, then there will be a corresponding microbial attack on our immune system, right? So first line of defense is all about preventing pathogens from getting a foothold in our body, right? The second line of defense is all about making sure whatever pathogen has come in, whatever pathogen has gained a foothold, it does not gain any more foothold than it already has, right? So I'll answer this one. Once the bacteria are within or have access to the living tissues of your body, but before cells are aware of their presence, bacteria will encounter chemical barriers, right? As part of the first line of defense, there are three barriers that a microorganism must get past. One is the physical barrier, the physical barrier that actually stops them from getting inside. Second is a chemical barrier. And then third is the microbial barrier. When we're talking about chemical barriers, we're talking about places where there are certain chemicals involved to prevent microorganisms from getting a foothold. For example, what is in your tears and saliva that acts as a chemical barrier? Can I get Janita? Lysozymes, right? That is the enzyme, lysozyme, and lysosome is the organelle responsible for the production of that enzyme. So lysozyme and lysosome are different. You guys need to make sure you know the difference as well. But yeah, basically, that's one of the examples, right? The lysozymes in our tears and our saliva were very strong, very capable of breaking down bacteria. Another one is also the stomach acid, right? If you've ever wondered when you eat something dirty or something that's fallen on the floor, the reason why you usually don't get sick is because your stomach acid is so strong that any bacteria or any microorganism that gets through your internal defenses will actually get destroyed at the stomach due to the strong stomach acid. So, like I said, today's lesson, we're going to focus on looking at the second line of defense, which is a non-specific response to any pathogens as well. So why is it called non-specific? It's in relation to the fact that it is not targeting any specific pathogen, right? It does not know anything about the pathogen at this stage. It is still just doing a broad response, right? Basically, it's the equivalent of me just trying to deal with the issue in the same way I would deal with any issue, right? The immune system is doing the same things it would for any of the pathogens because it is not aware of what the pathogen exactly is. It is only aware that it is something dangerous and something it needs to deal with. But other than that, it doesn't know any other details. That's why its response is still so non-specific, right? You will also look at the different types of leukocytes, which are white blood cells that are that have a role in the second line of defense. We also describe the function of different components of your second line of defense, and also the things that also happen during the second line of defense, which is, for example, fever and your inflammatory response. So you guys need, for this topic, you definitely need to know all the leukocytes and what they end up actually doing, right? All your macrophages, neutrophils, dendritic cells, eosinophils, neutrophil cells, mast cells, and complement proteins and interferons. You need to know what each of those are and what their function is. Okay, and you also need to know what the steps in the inflammatory response are related to. So let's start off with talking about the first line of defense. Like I said, the first line of defense, the main purpose is to prevent pathogens from getting a foothold in your body. But that can always be broken if you get a cut in the wound or you basically are getting exposed to a lot of microorganisms. Somehow or rather, they will get a foothold, right? The second line of defense is going to get activated as soon as that happens. So as soon as any pathogens make contact with our cells, they will actually trigger a response from our cells that actually act as an alarm bell that causes a lot of immune cells to start getting triggered as well, right? So the main player in terms of the immune system is called the leukocytes, right? White blood cells in definition. Okay, white blood cells is what they are meaning, but they are still not targeting anything about the pathogen, right? They are still part of the innate immune system because they are not specific. All they know is that an enemy has been found. We need to go kill that enemy, right? The same response, no matter what the pathogen is, be it a virus, be it a bacterial infection, be it fungal infection, it will still trigger the same downward responses that we're going to see right now, right? So when thinking about our defense, close enough, it is a non-specific response, right? The first and second line of defense are non-specific because they're not specialized towards any pathogens, right? They're not specialized towards viruses or bacteria or fungal. They are just doing the same thing for all pathogens. And the purpose of the second line of defense is to basically combat any pathogens that have made it inside and ensuring that they don't spread unnecessarily. Okay, they work in a combination with cellular responses and also non-cellular responses. The cellular responses is talking about the activation of these leukocytes to specifically target the pathogen, right? They will all go towards the pathogen, doing their different things, making sure that they are keeping the damage to a minimum. And then your body also triggers non-cellular components to have a role in the second line of defense, and that includes interferons, complement proteins, and the initiation of a fever, right? The initiation of a fever is a much more what's the word I'm looking for? Involved process that involves your body producing a bunch of triggers and responses for you to actually initiate a fever, but it is also still considered being part under the second line of defense. So let's talk about the different cells in your second line of defense. This is specifically the cellular components of your second line of defense, and that includes stuff like your neutrophil, your macrophage, your dendritic cell, your natural killer cell, your eosinophils, and your mast cells. Okay, each of these cells are different in their structure, their shape, and also somewhat in their function, especially sorry, these three, they have very similar functions. What these bottom three have very different functions between them, right? So looking at the first three, we have your neutrophil, your macrophage, and your dendritic cell. You can see that there's one commonality between the three of them, which is that all three of them are called phagocytes. Okay, what is a phagocyte? It is basically a class of cells that are able to engulf their prey. Engulf seems like a very involved word, but let's see. So if you need to know what engulf actually means, you just watch this video. This is a phagocyte right here. The colored ones are your pathogens. Observe what happens to the thing right there. Yeah, exactly right there. That is engulfing, right? It basically just took it into itself by swallowing whole. And how it does this is basically, if you looked at it in a cellular level, this is what you would be seeing, right? Essentially, you can see a part of its body extending over the pathogen, encompassing the pathogen, putting the pathogen within a vesicle, and then exposing the pathogen to lysozymes, right? The enzyme that will break down pathogens. And when that is over and done with, it will digest the pathogen and then release it in a process known as exocytosis. But essentially, it swallows, digests, and excretes the pathogen. The simplest way you can remember it, right? So the process by which it consumes and destroys the foreign dead material by engulfing them, that is called endocytosis. But the process by which it is actually just engulfing it, that is called phagocytosis. So it's more important that you guys know the word phagocytosis more, because that process will be questioned in the exam, and you need to know what that means, right? So once they've been swallowed, the lysozymes will release lysosomes will release lysozyme, and they will fuse and destroy the swallowed matter. Any questions here? So this is the three cells that can actually do that, right? Your neutrophils, your macrophages, and your dendritic cells. They are the three cells that are able to do this. But even among those three cells, there are two cells that are more in common than the third one, which is that macrophages and dendritic cells are antigen presenting cells, and they are also phagocytes as well, right? So neutrophils are only phagocytes, but macrophages and dendritic cells are both phagocytes and also antigen presenting cells. What does that mean is that when they do phagocytosis, your neutrophils and your macrophages and your dendritic cells, when they do phagocytosis, they don't just destroy, break apart, or digest the pathogen. They will then later store some of the remaining pathogens and present it as a antigen on their NHC class 2 marker, right? So if you guys don't remember your NHC class markers, let me give a quick introduction about them, right? So in our body, for our immune system to know the difference between our own cells and different cells, they have antigens on them, right? Antigens are how the immune response knows one cell from another, and to know that whatever cell our immune system is looking at is part of our own, they look for a specific type of marker called the NHC class 1 marker. Okay, this is a self-identification marker for our own cells, right? All of our cells have the same MHC class 1 marker, so all of them will be recognized as being our own cell. But at the same time, if we get any cell from a different person, let's say you get blood or you get an organ from someone else, their cells will have a different MHC class 1 marker. So they will be then found out as being different cells from different people. So they will trigger an immune response unless we make sure that the antigens are the same and stuff like that. So that is our MHC class 1 marker. It's about self-identification and it's only present on cells that are self, basically our own cell. But certain immune cells, like your macrophages and your dendritic cells, they also have a second marker called the MHC class 2 marker. This marker is specifically for presenting antigens to other immune cells, right? This will later come on very handy and it's part of the adaptive immune response. They are basically showing the rest of the immune system, this is the enemy. Make sure you memorize that this is the enemy by showing them antigens of the pathogen on their MHC class 2 marker, right? This will later come into hand when we're going to the third line of defense, which is a specific immune response, because they now know which pathogen they're working with. Because these cells are presenting the antigens, and those antigens are going to be processed, and then your body is going to know how to fight off that pathogen because it knows what pathogen it is dealing with as a result of these antigen presenting cells. Okay, anyone confused about what I'm explaining here? Everyone seems so cool, right? So they are only found, these MHC class 2 markers, they are only found in cells that are involved in the immune response, immune system, such as macrophages and dendritic cells, and they will basically move around in your immune system until they find the cells that they need to show this to, at which point they will trigger those cells as well. Okay, so this presenting antigens is related to the third line of defense, which is the activation of your adaptive immune system. So this will come in handy when we're doing a third line of defense. So now that we've covered the cells that can do phagocytosis, let's talk about the other cellular components of your second line of defense. The first one we're looking at is your natural killer cells, right? So these are basically search and destroy systems within your body that are looking for damaged or infected cells, right? In your body, there are a lot of things that can happen to damage a cell, right? Improper splitting when the cells were reproducing, that could cause them to be damaged. Or if it has experienced mutations becoming a cancer, that is also another way it can be damaged. Or possibly it's gotten infected with a pathogen, a virus of some sort. So that these are all things that can happen to our regular cells, and that makes them dangerous, right? For example, if we're dealing with a cancer, if a cancer isn't dealt with immediately, it can actually start to what's the word? Reproduce uncontrollably, becoming an actual malignant cancer. So it's very important that your body is able to recognize and destroy infected / damaged cells whenever they occur, right? And this only works due to a simple system that has two types of simple cell that has two types of receptors. One is called a killer inhibitory receptor, and the other one is called a killer activation receptor, right? So even though the names sound a bit confusing, a killer inhibitory receptor, basically an off switch. That off switch is different from a killer activation receptor, which is the on switch. They're not the same. They're not triggered in the same way. They are both going to have to work together to actually give a signal to the cell to kill or not to kill it, right? So talking about the inhibitory receptor, what happens is that it looks at the infected, damaged or sample cell, and it looks specifically for their MHC class 1 markers, right? The self-identification antigens that are used by your own body to show that your cells are actually your cells. This inhibitory receptor is looking for those markers on any cell it's analyzing, while the killer activation receptor is basically looking for signs that that cell is undergoing cellular stress. So cellular stress is happening whenever the cell is either doing something it's not supposed to, or it is undergoing some kind of a cellular process that is very demanding. Okay, then this is more in line with a cell being infected or damaged or cancerous. So it's basically looking for the signs that it could be infected or cancerous, which is the whole cellular stress thing. So two things, right? Killer activation, killer inhibitor. The killer inhibitor wants to find a reason to inhibit the killer. The killer activation wants to find a reason to activate the natural killer, right? That's why they're doing very different things. So this natural killer system is only activated on the case that both of them are supporting it, right? If the killer inhibitory receptor is saying that there's a lot of MHC1 class markers on this cell, then it won't trigger the kill switch, right? If it says there are very little MHC1 class markers, that is a go from the killer inhibitory. And then if the killer activation receptor also says that this cell is undergoing cellular stress, both of them are in agreement, and at that point, they will basically trigger cell death by a process called apoptosis. Right? Cell death is only initiated in infected or abnormal cells with missing MHC1 classes, MHC class 1 markers, and where the killer activation receptors are activated, right? So two things need to happen for any cell to be killed by the natural killer cell. One is that they should not have a lot of MHC1 class markers so that the killer inhibitory receptor is not activated, right? It really wants to inhibit this cell from killing anything, and if it is activated, then nothing else will happen. So only gets activated if there is enough MHC1 class markers. So if it finds that there are not enough, then it won't get activated. So the next step is looking at the killer activation receptor. That is looking at whether the cell has molecules which appear on cells undergoing cellular stress. If both of those are present, this one doesn't get activated and this one does get activated, then that will trigger the kill system, causing it to kill the pathogen, or sorry, kill the infected cell. Any questions here? All right, hopefully everyone understands. All right, moving on is the different types of cells, right? So next is talking about your mast cell, right? So remember that when I said that when you get a cut, a part of your body gets triggered and basically acts as an alarm, triggering your immune response to respond there. That is what your mast cells act to do. They basically release histamine by degranulating when they detect antigens or allergens are present. Okay, this histamine is imperative in triggering a inflammatory response, which is a type of response that basically forces down a fluid to one section of your body, and in turn it brings along the immune system and also a way for you to deal with whatever cut or whatever injury you've gotten by plugging it up, right? So if you've ever wondered what degranulating means, it's this action where an antigen comes into contact with the mast cell, and as soon as it does that, the mast cell literally just ceases to exist and releases all the histamine it has. Yeah, that's degranulating, right? It just releases all the histamine that it has at that very stage, right? So mast cells are mini alarms to your body that basically signal your body, okay, here's where something has happened. There's pathogens being detected here. Everyone come here and defend the body, right? They need these initial alarms, or else your body won't know where it's getting punctured, right? You might get a cut on your leg, you might get a cut on your arm. but the mast cells at those points releasing that histamine is what triggers your immune response to be so rapid at that site. All right, moving on to the next cellular component is your eosinophils. They are basically chemical warfare experts. They use stuff like DNAs, RNAs, and also protease to help destroy invading pathogens. And they basically handle the cells that cannot be phagocytosed, 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 and they basically release all the chemical mediators when they are exposed to the pathogen. They are basically suicide bombers that carry chemicals, and they do not like it when pathogens are invading. So they release these chemicals as a way to stop them or to basically kill them as well. All right. So now that we've talked about all the cellular components of your immune system, let's focus on the non-cellular components, which are not exactly cells. They are either proteins or they are an entire cascade response, right? So talking about your molecules, first one we're looking at is your interferons. Interferons are basically a molecule released by a cell that has been infected by a virus. They basically act to signal the neighboring cells to be less susceptible to the viral infection. So, essentially right here, right? So we have our regular cell, it's been infected by a viral infection, right? That viral infection basically forces our own cell to become a protein manufacturing machinery, and it starts to make that cell reproduce more of that virus. At that stage, this cell is releasing something known as an interferon, which is basically a signal that says, as a virus inside me, save yourself, right? When those interferons are exposed to different neighboring cells, there are three things that can happen. One, it signals the neighboring unaffected cells to destroy RNA and reduce protein synthesis. Why is this important? Is because the virus acts to control the cell by making RNA and also controlling its protein synthesis. So the cell will go into hibernation mode and not really accept the virus anymore. Okay, second thing is that it signals neighboring infected cells to undergo apoptosis. Apoptosis is signal cell death, and interferons can sometimes even do that, right? Especially when it's neighboring infected cells, they trigger those cells to undergo cellular death. Or third one is to activate your immune response as well. So in the same way that histamines activate an immune response, interferons are capable of doing somewhat the same thing as well. Moving on is your complement proteins. Okay, complement proteins are actually quite dense of an idea when you guys try to absorb it. Essentially, in the most simplest way you can understand it, complement proteins are proteins that are obviously present in your body, and there are billions of them in your body right now. Okay, there are sheets of them in your body. These are basically, how to put it, almost like Legos floating around in your body that wait for something to trigger them, at which point they build into something bigger, right? They build into something bigger for a specific purpose. So, for example, there are three ways that a complement system can work to a pathogen. One is opsonization, the other one is chemotaxis, and the last one is lysis, right? These reactions, these cascade systems, complement cascade systems, are part of why pathogens cannot get a strong foothold in your body. Without the complement system, your body is also heavily defenseless against pathogens, right? So if you want to get an understanding of what each of these are, just give me a sec. We'll get into that. But the last non-cellular component of your second line of defense is your fever, right? The fact that you get a fever is a programmed thing by your own body. Your body wants you to get a fever for a few reasons, as we will discuss very shortly. So essentially, let's talk about our complement proteins first, right? So when we're thinking about a complement protein, they are basically, like I said, Lego pieces that are floating around in your blood, billions of them. And when they are triggered by a pathogen, they basically build into something bigger or build into something more useful, right? For example, when a pathogen has triggered the complement proteins, one of the things that can happen is opsonization, which is essentially marking that pathogen for your immune response. Basically, here's the pathogen. That's it. Like it's been marked. The pathogen has been marked such that the proteins stick to the outside surface of the pathogen and make it more of an obvious target for the immune system, right? Basically, like I said, it's just a marker for your immune system, right? So anytime the immune system is around, it will immediately know this is a pathogen I need to take care of by due to the fact that the complement proteins are stuck to the surface. Second is chemotaxis. This is where it becomes an attractor for the immune response, right? Not just something to allow them to identify the pathogen faster, but it actively attracts the immune response to the pathogen as well. How does it do this? Is that it gathers near the pathogen and essentially just attracts the phagocytes to it, right? They act like a magnet, drawing in the phagocytes to them, at which point they will get exposed to the pathogen, right? So let me go through the first one again. Like I said, the first one, opsonization, is binds to the surface of the pathogen. They act as a marker that basically makes it easier to identify for the immune response. That is the first one. The second one on the second one, however, is more about becoming a point of attraction, draw in the phagocytes such that the pathogen gets revealed. Right? The pathogens here, the complement proteins are surrounding the pathogen, and then they basically attract the phagocytes close to them, and at which point the phagocytes will go to town on the pathogen. And lastly is the fun one where it's lysis, which is where the complement proteins work together to become a hole puncher in the pathogen itself, right? What do I mean by a hole puncher? It basically literally punches a hole in the surface of the pathogen such that excessive fluids will go into the pathogen, causing it to explode. The equivalent would be basically you getting overinflated by air and then you exploding, right? That's what's happening to these pathogens right here. As soon as they have a hole in their membrane, excessive amounts of liquid will flow in, and that will cause the cell to burst and die. This only happens because the complement proteins, like I said, they're Lego pieces, they build to something bigger. That's what's happening here, right? This structure here, the membrane attack complex, is a result of multiple complement proteins forming together to become into a complex. And lastly, we have your fever, right? Your fever, like I said, is a temporarily increased body temperature by your body for the sake of combating a pathogen. Two reasons why they do this, right? One, pathogens hate it. Pathogens hate the hot environment, right? Because like any organism, they have a narrow window of optimal temperature for their day-to-day processes. So pathogens don't like the fact that the temperature suddenly went up because that's probably not a temperature that they are optimal for, right? This is not always going to work though. Even some pathogens might love it that it's getting warmer because their optimal temperature is different, right? So this is not a one system defeats all. It's more of a generalized response that is gonna benefit most of the time. Okay. The second reason is also because it activates your immune response and certain proteins to be produced such that it acts as a boost of strength for your body's defenses. Essentially, your body, your immune response especially, works better in a slightly hotter environment. As such, they actually help the immune system by having a fever. But by nature, we don't like fevers, right? We do whatever we can to stop them. We take medicine, paracetamol, uh, ibuprofen, be whatever. You're basically also putting a stop to this, right? These two things, they are have an advantage when trying to defend your body against pathogen. But by dealing with the symptoms of a fever, when you do have a fever, you're also circumventing the actual positives of that fever, right? So why would we do that? Why would we circumvent the natural system that our body has to combat a pathogen? Essentially, fever can be harmful to us as well if it is present for a prolonged time. Our body doesn't really think about these things, so we need to basically take that into consideration. All right, watch this short video on fever.
What now? Che what? Ricordo, vestiti o similari, TV? What Every animal or microbe has a temperature range that's ideal and one that's stressful but survivable for a while. Your ideal temperature is where your cells work best, where their internal machinery is the most efficient, and the animal as a whole the best adapted to its niche. Humans are warm-blooded animals, and our bodies expend a lot of energy to keep us around 37 degrees Celsius or 98.6 degrees Fahrenheit, which seems wasteful, but this may actually be a defensive adaptation. Our temperature makes us almost entirely immune to one of the worst killers and parasites, fungi. Most colder animals and their insides are infected by them, but you are just too hot. Which brings us to fever. For any microbe that wants to infect you, your body is a world they want to conquer. Fever is defensive climate change, pushing an invader outside its ideal temperature range and making the world horrible. It evolved at least 600 million years ago and is widespread. Most animals increase their core temperature when they're sick. Fish swim into warmer waters, lizards bathe in the sun, bees heat up the air inside their hive. But you, warm-blooded mammal, you have way more drastic options. Let's make you sick and see what happens. When your blood turns into lava, you're invaded by bacteria and viruses at the same time. The invasion is powerful, and you need to slow it down as fast as possible. Fever is part of your first line of defense, triggered by a diverse group of chemicals called pyrogens, the creators of heat. They float away from the battlefield and pass right into your brain, where specialized receptors pick them up and crank up your internal thermostat. First, you begin to shiver. Your skeletal muscles contract really quickly, which generates a lot of heat in your core. At the same time, usually the blood vessels near your surfaces contract and prevent heat from escaping through your skin. Your skin cools down while your insides burn. Fever is a systemic body-wide response and is a serious energy investment for your body. You burn about 10% more calories to stay alive for every degree centigrade your body temperature rises. Fever is also a strong order to lay down and rest, to save energy and give your immune system time to fight. Back to the battlefield. When a bacteria entered your body, they tried to be stealthy, but now they've switched into high production mode. Their goal is to multiply as fast as possible, which means they need a lot of resources and are highly stressed. Imagine running a marathon while eating a succulent Chinese meal or giving birth. The last thing bacteria need right now is more stress. So your immune system tries to stress them out as much as possible by ordering inflammation, which floods the battlefield with fluids, attack proteins, and soldiers. Pretty stressful. Fever is even more stress. For the bacteria a moment ago, the temperature range was pleasant. Now the world burns. Heat can cause their organs to break and the membranes to rupture, damage their DNA, and diminish protein production. They are seriously suffering from the heat. Why doesn't this affect your cells? It does. All of this is stressful for your cells too. Virtually every system and organ of your body works worse during fever, except one, your immune system. Neutrophils are recruited faster. Macrophages and dendritic cells are better at devouring enemies. Killer cells kill better, and so on. And fever animates your immune cells to gobble up the critical resources your enemies need, like iron, glucose, and glutamine, turning the battlefield into the food desert. The viruses that infected millions of cells are doing even worse because they're also very sensitive to heat. For example, the rhinovirus that causes the common cold can only infect your respiratory tract because it is significantly colder than the rest of your body, even without fever. The heat is also really bad for the millions of cells that are infected by viruses at this point. They are working super hard producing viruses, which is pretty stressful. As the heat becomes too much to bear, the super stressed cells panic. As their internal machinery is breaking and failing, they quickly produce billions of heat shock proteins, or HSPs, that start repairs, keeping them alive. But this is a trap. Even your healthy cells produce HSPs to deal with the heat, but if a cell makes too many of them, this means it's more stressed than it should be. And if it's too stressed, something is wrong and it should be killed. So your natural killer cells and killer T cells are activated and attracted by HSPs and start killing infected cells and all the viruses inside them. By trying to protect themselves, infected cells are calling out to be destroyed. But if fever is such an effective weapon, why don't your enemies adapt to it? Is it still viable in so many different animals after hundreds of millions of years? A wild reason is that fever actually might outsmart evolution. If your enemies survive fever long enough, natural selection changes them. The individuals that are better suited to deal with heat reproduce more. After a few days, they've adapted. But this becomes a handicap because the next step is to infect new victims in new bodies, and now healthy humans are too cold for them. Not impossible to infect, just harder. And the heat-resistant microbes now compete with their cousins that like it colder and have an advantage infecting healthy hosts. This creates an evolutionary dilemma without a perfect solution. To circumvent this, serious pathogens like measles use hit-and-run tactics. The measles virus is the most infectious right before your fever hits with full force. It's brutally beaten back once your full immune response shows up, but by then, the damage is done. Fever is an effective part of the puzzle of your immune system, helping to attack and stress your enemies from as many angles as possible. But if fever is so great, why do we stop it when we're sick? Should you fight fever with medications? We think it's normal to have magic pills, but relatively harmless over-the-counter pain medication like aspirin or ibuprofen only became cheap and widely available in the last century or so. Going to a pharmacy to get something for your headache is extremely new in human history. Pain feels bad, so we've gotten used to stopping it when we feel it. If you're sick, you're supposed to feel a reasonable amount of pain, so you lie down and save energy. This is not a bug, but a feature of your immune system. But pain and fever are closely connected, and over-the-counter pain medication like ibuprofen and paracetamol also work against fever. Especially in children, fever is often suppressed by worried parents or doctors, sometimes because they think fever itself is the disease, or they're worried that it can do long-term harm. In general, it's fair to say that for temperatures below 40 degrees Celsius or 104 Fahrenheit, fever is not dangerous and doesn't need to be treated. Of course, there are also patients that should not have fever, like pregnant women, seniors, and seriously weakened patients. For them, the extra stress may be dangerous. Fever over 40 degrees Celsius is dangerous to anybody because it's most likely caused by your internal heat monitor failing. Things get more complicated in serious disease territory. We also have evidence that for some diseases like influenza or chickenpox, anti-fever drugs do not help you to heal faster. But we're also running into ethics problems here that make clinical trials difficult. In one study, doctors gave strong anti-fever treatment to critical care patients, but had to stop after mortality shot up. Overall, we have strong indications that more people may survive serious infectious diseases better with a fever. And there is very little clinical evidence that stopping fever leads to better health outcomes. But there are important exceptions like neurological injuries and stroke. We definitely need a lot more research. So, should you fight fever? Well, speak to your doctor and don't listen to internet videos. But this decision is really about payoffs. If a fever is not dangerously high and you can bear it, you're supporting your defenses and may even get healthy a bit faster. But if you feel really bad and are healthy in general, against pain and fever will make you feel better quicker at the cost of a slightly less effective immune defense. However you decide, the next time you're burning up and feeling bad, you can rest easy in the knowledge that your enemies are having a much worse time than you.
The good guys do most of the use of what now? Nearly start 60 like sugar.
All right, so essentially that's what fever is for, right? Fever heats up, increases the temperature of your body, and that has two effects. One is that it becomes a unsuitable environment for whatever pathogen that's invading, right? Remember, all organisms have a optimal temperature that they operate at, and if you bring any organism out of that optimal temperature, they will be under stress, right? That stress comes with a price. So the pathogens have a negative. Another reason why it also happens is that even though the rest of your body may not like that increase in temperature, your immune system loves it. Your immune system operates at a higher efficiency at the high heat. In fact, that is actually why one of the major reasons your body even does a fever. So there's twofold for the presence of fever. Make sure you remember that. Okay. Now, what we're going to look at is basically the steps involved in your inflammatory response, right? Inflammation is a process in which a few things happen, and it's always triggered by the break of the first line of defense. So a perfect example, you just got a cut on your hand. That cut exposes your internal body environment to the external body environment where there are a lot of different pathogens. So whatever opportunistic pathogens that were present on your skin at that point would enter into your internal system, and they will get exposed to all your stuff on the inside. This is when a few things will happen, right? This is when your mast cells will get triggered by the antigens on the pathogens, right? They will then release a bunch of histamine, right? Remember the histamine that mast cells release? That is what triggers the whole inflammatory response in the first place. It has to be started by the mast cells releasing histamine, and then that histamine triggering an inflammatory response to that region. When that inflammatory response has been triggered, what are the things that happen? One, blood flow to an injured area is increased. So it's increased, meaning that the amount of blood flowing to that region will be increased. Think of it like an emergency system that forces more fluids into that region so that more white blood cells and more platelets can arrive at the scene. Second thing is that there's a greater number of immune cells arriving at the site of incidence. Yeah, that is a downstream effect of the more blood flow. Third thing is that the site of incidence is cleared of any debris. Obviously, when there is an injury that occurs, then not only is it the pathogens get in, but there's also a lot of damage and destroyed cells in that region, right? Because whatever cells that were punctured, they are all destroyed. So there's a lot of debris just lying around, right? Debris of our blood vessels, debris of our cells, debris of whatever that was in the way when that needle poked through, they will just be sitting around there. They need to be cleaned out if that wound is to be shut clean, right? And then lastly is the repair of the site of incidence due to the action of platelets. So when thinking about the steps involved in the inflammatory response, there are three distinct steps. One is initiation, the second is vasodilation, and then the third is migration. Each of them are specific in what is happening, right? The first one, initiation, right? Like I said, your mast cells degranulate, release histamine at that very stage, right? This is the initiation of the inflammatory response. and that happens due to the fact that histamine is being released by the mast cells. A few other things is also happening here, where damaged cells also release the signaling molecule known as cytokines, where it is basically signaling the immune response as well. But what happened is that the first thing always is happening first, right? The first line of defense has been broken, and that brings in pathogens, and those pathogens trigger two things. One is the production of cytokines, and the other one is the production of histamine. Okay, histamine is produced by mast cells, and cytokines are just produced by the damaged cells. Moving on is the second step, which is vasodilation. Right? This is the step in which excessive amounts of fluid is emptied into that region. And how does that happen? Is basically the histamine from the mast cells cause blood vessels to dilate, making it permeable to that region. Right? Basically, blood vessels, they can dilate or constrict, allowing them to let more blood come out or stay in. When these mast cells are releasing histamine, they trigger the blood vessel to vasodilate more, basically expand more, allowing there to be more pores for the blood to flow out. Right? This increases the blood flow around the swelling, resulting in swelling and redness. So anytime you guys get a cut, the reason why it gets red and hot is because it is undergoing the inflammatory response. Okay, so now that everything is in place, the third step, which is migration, is basically when the inflammatory response brings everything that is needed to that specific location, right? The cytokines from the damaged cells tell phagocytes to come to the damaged cell. Basically, as the phagocytes are flowing through the blood vessel, any cytokines that go into the blood vessel will will trigger the phagocytes to actually come out of the blood vessel into the damaged region, because they have been triggered by the cytokines to actually come fight the war. Second thing is that complement proteins also help to attract pathogens, to destroy them. So this is the complement proteins also coming in to do that thing. And then thirdly is the platelets coming out to basically clot the wound and make sure it does not still allow any more pathogens to come through, right? So three-step response. One is initiation, second is vasodilation, and then third is the migration. The first one is about how is it that the inflammatory response is triggered. It's triggered due to the fact that something has broken the first line of defense, bringing in some pathogens and some debris. Those pathogens trigger the mast cell to release histamine and trigger all other cells to release cytokines. Then the second, which is vasodilation, the mast cell releasing histamine causes the nearby blood vessel to vasodilate, which is basically expand in size, allowing more blood to seep out, to flow out into the damaged region. With more blood comes more immune cells, right? Your phagocytes, your natural killer cells, your eosinophils, and so on. They will all come to that region because of vasodilation, bringing them in, right? As soon as that happens, they will basically get triggered to combat the pathogens. They will also clear the debris as much as they could. And then lastly is when they do all the things that they are expected to do, while also getting help from complement proteins, and platelets will finally come in and seal the wound through coagulation. Sorry, what platelets do that? Basically, they just clot through clotting. All right. So what happens after all this war has gone on is basically the end result of your inflammatory response is basically a battlefield with hundreds, if not thousands, of dead pathogens and also dead immune cells. Right. This battlefield of casualties are later basically put together and comes out as a pus-filled pimple. So next time you see a pus-filled pimple, just know that your body has fought a war of pathogens off, and is basically presenting the remains in that tiny sac right there. Any questions? Right, let's watch a short video talking about this, shall we?
Get KFC delivers for free on menu bar. Use code FREEDOM100.
I thought it's so cheap.
The good guys are always the only one now. If they start saying like what should I need before you? Stop this. Did you come to say you?
It's been a normal day when suddenly the world explodes and an asteroid rips the sky open. Countless alien life forms invade, ready to destroy cities and infrastructure and eat civilians. Or this is what your cells experience. You look at your bleeding thumb that you just cut on a dirty twig in the park. How annoying. But inside the wound, a horrible catastrophe has happened. There are dead cells and blood and dirt everywhere. Even worse, countless bacteria invade the warm caverns between your helpless cells to explore their new home, steal your resources, and poop everywhere. Immediately, the first stage of your defense kicks in. The cells that survive the impact or are hurt or dying scream in panic, releasing an onslaught of chemical alarm signals that awaken your immune system. The first cells to show up are macrophages. If an average cell were the size of a human, a macrophage would be the size of a black rhino. A stoic cell in principle, but you wouldn't want to annoy it. Bacteria do annoy them. Within seconds, the large cells attack and begin killing them without mercy. They stretch out parts like the arms of an octopus and grab the bacteria to swallow them whole and digest them alive. A macrophage can eat 100 bacteria before it's exhausted. But there are too many enemies, so the macrophages call for reinforcements. In your blood, hundreds of thousands of neutrophils pick up their signals and move to the battlefield. Neutrophils are intense suicide warriors that only live to kill. They're so enthusiastic about killing that they kill themselves a few days after birth, so they don't have time to accidentally destroy your body from the inside. As soon as neutrophils arrive, they begin vomiting deadly chemicals at bacteria or devour them. They are so careless in their attacks that they are causing real damage to your own cells, but collateral damage is not their concern now, or ever. Some neutrophils go so far to push their suicide button and explode, casting wide and toxic nets made from their own DNA, filled with deadly chemicals that trap and kill bacteria. Sometimes they can continue fighting after that, even though they're sort of dead already. This is how much fun they have killing. While the battle rages, your blood vessels let fluid stream into the battlefield like a dam opening up towards a valley. You notice this as inflammation. Your thumb swells up a little and gets red and warm. The fluid brings the silent killing into the battle zone, millions of complement proteins, a sort of automated liquid weapon that stuns and kills bacteria by ripping holes into them. We made a whole video explaining them in detail. We're reaching a crossroad now. If things go well, your first line of defense kills the invaders quickly. But sometimes the enemies are too strong and would overwhelm your defenses eventually, which means certain death for you, the human. This is the hour of the dendritic cell, your immune system's intelligence officer. While your soldiers were bashing in heads, it was collecting samples by ripping bacteria into tiny parts and covering itself in it. Like a soldier decorating itself in the guts of a dead enemy. The cell leaves the battlefield and enters the superhighway of your immune system that connects all your tissues with your immune headquarters, your lymph nodes. The dendritic cell coming from the battlefield is looking for a helper T cell, which is a sort of all-purpose commander cell within your immune army. But not any helper T cell, one that happens to have just the right weapon for the bacteria.
All right. Everything that comes after this is part of your adaptive immune response. Okay, everything we looked at in this video before this, that is part of your non-specific, specifically your second line of defense. Now, what's going to happen is your body is going to recognize what that pathogen is, right? Who that pathogen is, whether it's a virus, bacteria, or so on. And it's going to try to find a specific immune response that is targeted towards that pathogen.
Infected your wound. So it goes around and rubs itself, still covered in bacteria parts, against every helper T cell it meets. Most T cells are a bit disgusted and not interested, but after a few hours, something clicks. A helper T cell recognizes the bacteria parts. This cell is the weapon that's needed right now. The dendritic cell is overjoyed and activates the helper T cell. Okay, wait.
Wait indeed.
You can't be saying the good guys do it's Psalm 60 like what should I be for you? So they said it's like 79 each team. 70% of HP computers. This is
How come your immune system has a cell that has a weapon against the specific bacteria that infected you? Well, your immune system has a perfect weapon against every possible disease in the universe, against the Black Death, the coronavirus, or an infection that will emerge in 100 years on Mars. We'll talk about this a bit more in the next video because it's very complex. So for now, just know that you have billions of unique helper T cells that each have weapons against every possible enemy. After the right T cell is activated, your second line of defense awakes and rises like a teenager that needs to get up on a school day, very slowly. Your heavy weapons are incredibly effective, but they're not fast. The activated helper T cell begins to clone itself over and over again. 1 becomes 2, 2 become 4, until there are thousands of them. Now they split into 2 groups. The first group quickly moves to help out your soldiers. At the battlefield, things are getting out of hand. A tired macrophage is ready to give up. After fighting for days, it just wants to go to sleep, like many of its buddies have done already. But now the helper T cells arrive. One of them comes to the tired macrophage and whispers something using special chemical signals. In a heartbeat, the demoralized soldier feels fresh again. But there's something else, a hot, white anger. The macrophage knows what it needs to do. Kill. Invigorated, it throws itself against the enemies once again. All over the battlefield, this begins to happen. Meanwhile, the second group of helper T cells was working on activating another line of defense, B cells, your antibody factories. Antibodies are protein superweapons that look like tiny crabs with 2 pincers to grab enemies. Just like the helper T cells, there are B cells in your body that are able to make just the right antibodies for every possible enemy. And the helper T cell is looking for exactly these B cells. After a day or two, the right B cell is found and begins to clone itself. As soon as enough clones have been made, each B cell begins pumping out up to 2,000 antibodies per second. About a week after you injured yourself and bacteria invaded, your second line of defense finally arrives in full force. The tiny army begins to saturate the battlefield, pinching and stunning desperate bacteria. The antibodies clump them together and make them unable to move or fight, while your soldiers massacre the defenseless victims. The tide is turning fast. As the last enemies are cleaned up, your soldiers realize they are no longer needed and begin to kill themselves to save resources. But not all of them. A few helper T cells remain and turn into memory cells. They will guard the tissue for years, making sure the same bacteria will never again gain a foothold here. Similarly, a few B cells will stay alive and keep producing a low amount of antibodies, making you immune against this bacteria, maybe for the rest of your life. One day you wake up and notice that the wound has grown over and left nothing but a faint red mark. You were completely unaware of the drama your cells had to deal with. For you, the whole ordeal was a slight annoyance, while for millions of cells, it was a desperate fight for life and death. But this is just the beginning of the epic story that unfolds inside you every day and is told in full.
Alright, any questions? Alright, essentially, just remember the cellular components of your second line of defense, non-cellular components. The cellular components, the top three, neutrophil, macrophage, dendritic cells, they are phagocytes, meaning that they engulf the pathogens to destroy them. Macrophages and dendritic cells, however, are antigen presenting cells that are related to your adaptive immune response. Natural killer cells, they trigger cell death if it is a damaged or infected cell. Mast cells release histamine, eosinophils are chemical regulators, and so on.